Thus far, we have gotten from beginning beginning of life. So we got original organic precursors. Now we've built them into polymers. The next step in order to get life is to enclose them in some sort of protective barrier. That's the goal now, protective. We got to get these cell-like structures. At this stage, what we're building is called a protobiont. So proto is, I think, first. So first bio is first life. We're going to get a bunch of prebiotically produced molecules. So a bunch of these polymers coming together. They may have gotten a phospholipid bilayer. They may have gotten what's called a coacervate. But the big thing is they're going to have an inside and an outside. And having an inside and an outside is a big deal. You have an internal environment that you or that this thing will regulate and an external environment that it's reacting to. So we have an in and an out. That's big. The features could have been that it was a amphipathic. It could both repel and attract water at the same time. As long as you're able to separate the inside from the outside, that's all you need for a protobiont. Inside, you have an aqueous compartment that has some sort of macromolecules. Outside, you're existing in an aqueous world. Aqueous, of course, meaning water. The polymers on the inside were likely able to gain some sort of metabolic pathway to allow them to link to each other. It's within that enclosed space, within the inside, within that protected area, that these long molecules, these big polymers, began to enzymatically link other molecules to each other. And that was a big step, to take one molecule and connect it to another. It's like saying RNA, from random formation, randomly forming a piece of RNA, due to sheer dumb chance, was able to grab another piece of a nucleotide and attach it to itself. Is it possible? Yes. Is it likely? Not in the course of our lifetime. But you need to have that step. You have to have that enzymatic part to speed up reactions. We needed to get a chemical that acts as an enzyme, lowering the activation energy to build more macromolecules. Once you get that step, once you get enzymes built, these reactions happen at a much quicker pace, leading to growth. So we know what we need. We need an outer membrane, and we need a way to enzymatically link enzymes. So let's talk about that membrane first. Because once a protobiont gains the ability to grow and process materials, enzymatically linking things together, they get bigger and bigger and bigger until they split. A cell has a maximum size. You never see a single-celled organism that's the size of a person, because there's a lot of constraints on it. One of the constraints is the way the outer layers work. Have you guys ever blown bubbles before? You'll notice that bubbles can only get so big before they pop, or before they split into two bubbles. A cell will do the same thing. As it gets bigger and bigger and bigger, it will automatically pinch itself off into two distinct cells. It's a property of the phospholipids. When that happens, if we had molecules on the inside, some of the molecules would go in one bubble, some would go in the other, and boom, two distinct protobionts. There are two competing theories for what the first coverings were. One theory is the liposomes, which you guys know a lot about. One is coacervates. You may not know liposomes by the term, but you've studied them a lot. The other is coacervates. Talk first about coacervates. If you take a charged polymer, so DNA, a protein, we've got polymers, right? We were just talking about how we build polymers in hydrogen sulfide and such. Once you have those and you mix them with water, they're going to form structures called coacervates. So if you've got proteins, if you've got polypeptides, if you've got nucleic acids, and you just mix them up in water, they will begin to form these automatically that make little bubbles. It's because part of them are going to orient toward the water, part will orient away from the water. These bubbles are just empty cells called coacervates. If you were to trap an enzyme inside, those enzymes could have some primitive metabolic function. So that's one possible formation theory. If you took charged particles like polypeptides and nucleotides, nucleic acids, they can automatically form a coacervate with an inside and an outside. The other one are the liposomes. Liposomes are made of amphipathic molecules like phospholipids, where the head faces water, the tail away from water, and if it's big enough, you'll have a secondary structure on the inside. So you have a phospholipid bilayer. Do you guys remember that term? In which you get water inside, no water, then water outside. So those are liposomes. One type is obviously it's got an inside and outside. The other is the micelle. We're not worried about micelles. Mainly worried about liposomes. Inside and an outside. If you trap an enzyme in there that's able to undergo metabolism, now you can build more and more and more of these phospholipids or whatever, and it gets bigger until it splits. So you can have either coacervates or liposomes. Which are the ones that we currently see most often with living organisms? Liposomes. Liposomes are self-forming. That is, they do not require any energy to build. That's useful. You don't have to rotate them. They naturally congregate, naturally form these structures. And then you can use liposomes to form electrochemical gradients. That's where there's more of a charge on the outside than the inside, or vice versa, that will allow for flow of ions. And as those ions flow, it can create energy. Energy that's usable by any enzyme that's inside the liposome. So we've got liposomes and we've got coacervates. Liposomes are a likely hypothesis for the first model of how the cell formed, because if you were to take slightly moist clay, back to clay again, right? For some reason, clay keeps popping up. It's been shown to catalyze the formation of the liposome. If RNA is present, that is, if you have a piece of RNA, a nucleic acid, on the clay, a liposome naturally forms around it. So this facilitates the formation of protobionts. So far, so good? Questions, concerns? That's how we can get our membranes. So we needed these cell-like structures to form. They could have formed as either coacervates or as liposomes. They needed to form around some sort of enzymatic structure, which we haven't yet talked about, how we've got these enzymes, these new catalysts forming. So far, we know that if we were to use metal cations, if we were to use clay, we could build these things, but it takes time.